Hi everyone, we're so glad that you've joined us at the Center for Brain Health for our event. Let's get started with a brief video underscoring the importance of brain health for you and for society. After that, we'll introduce our speaker.
We work, we live, we innovate, and create. At the center of it all is your brain health. The ability to solve problems, think analytically, share empathy, and thrive.
We're trying to make brain performance really the next fitness revolution.
Scientific discoveries show that your brain can adapt and grow regardless of your starting point. The Center for Brain Health can show you how to transform your mind to work stronger and faster.
We learned from a national poll that 95% of people want to do something about their brains. The problem is they don't know where or how to start. At the Center for Brain Health, we have easy-to-use, scientifically proven tools to provide people a proactive path to do just that, to help people everywhere improve their quality of life through brain health. What's exciting is that it's easy to start, and starting is urgent because we are in a brain health crisis.
So how do you boost your brain power? How can better brain health help you focus on what's most important?
Brain training is a part of the way that we would distinguish ourselves from others. Quite frankly, I think it's going to be commonplace in businesses in years to come.
We are seeing an epidemic of mental health concerns. Stress and overload lead to depression, anxiety, and burnout. Our brains hold the key to navigate these hurdles. It's never too soon to start.
I think the greatest national security threat is pre-K through 12. If we don't take care of educating our young men and women, then we have to ask ourselves, where are we going to be in 20 years?
Research shows training middle schoolers how to think, problem solve, and innovate significantly improves academic achievement. Even more importantly, it enhances confidence and well-being for them to thrive throughout their lives.
It really helps the kid and it builds their stamina. I genuinely credit my success in the classroom from this program.
The key to the future is stronger brain health for individuals and communities. Ready to transform the world with us? Be a part of the brain health revolution. It starts here.
Okay, everybody. On behalf of everyone at the Center for Brain Health, welcome to the Frontiers of Brain Health, where we take a deep dive into the most exciting new developments in brain research. We'll have a few minutes at the end. So I'm going to be introducing Russ Poldrack momentarily, and we'll have a few minutes at the end to answer some questions. So for our virtual audience, please use the Q&A function. And for those of you who are in person, please remember that we are being recorded when you ask your questions. For those of you who aren't familiar with the Center for Brain Health, we are a cognitive neuroscience research center at the University of Texas at Dallas, and I have the distinct honor this morning of introducing Russ Poldrack. And Russ and I we were just figuring out about 20 years or so back. Now we're postdocs together at Stanford University. But first I want to say, that Russ is sort of a Lone Star State favorite son because he did his undergraduate degree at Baylor University just down the road in Waco. He went from there to the University of Illinois, where he did his doctorate with Neil Cohen. And then joined us at Stanford in 1995. He went from there to his first faculty appointment was at MGH and Harvard Medical School, moved there from UCLA, and then did a stint at UT Austin, actually, helping to rebuild their imaging center. So we had some things to commiserate over dinner last night about running an imaging center. And then went from there to back to Stanford as a professor, actually as the Albert Ray Lang professor, and he is also the director of the Stanford Data Science Institute at Stanford. And so with that, I want everyone give Russ a warm welcome and hear what he has to talk to us about today.
How's that? Good, cool. All right, thank you. Slides, okay. Well, thanks so much for the invitation. This is actually the first one of these types of talks I've done since COVID, which is fun. And so I was thinking about the last time I was here and looked back, and I gave the last talk here in 2009, almost 15 years ago. And it was about how we stop ourselves, and that's what my research was working on then. We still do a lot of research on that. But if you saw the announcement for my talk, I gotta turn that on. If you saw the announcement for my talk, you probably noticed that I'm talking about something very different today, right? I'm not going to tell you how any particular thing in the brain works. I'm talking about how do we change the way that science works. And so what happened, you might ask yourself, to cause such a shift in what I'm flying around talking about? And the answer is this. Over the last decade, we've seen what a lot of people call a reproducibility crisis. The idea is that we've realized that a lot of the ways that we went about doing science didn't end up giving us answers that were as trustable as we thought they were. And I would actually recommend Richard Harris's book if you want to learn more about this. He's a really good writer and does a good job of it. So I want to talk about what do we mean. We talk about a reproducibility crisis, what do we mean? And different people use the term in different ways, and I like this way of thinking about it. So basically, we can ask ourselves, are we talking about using the same data and getting the same answers, or different data, and using the same analysis or different analyses? And so I'm going to walk through each of these kind of different ways of asking the question, starting with what we call generalizability, the idea of if you have different data and a different analysis asking the same question, can you get the same answer? And another way to say that is, and I'm going to be focusing on brain imaging, are these, can we come up with the same conclusions given different data and different analyses? And I want to show you at least one example of a case where this seems to have not worked. This is, there's been a bunch of research looking at what differs in the brains of people who suffer from depression symptoms versus people who don't. This was what's called a meta-analysis, where people put together a bunch of different studies to ask, are there any kind of common findings across a bunch of different studies? And earlier analyses had suggested that if you put different studies of depression together, you could find kind of a common answer. But this study, published in 2017, done by the world's best group at kind of combining brain imaging studies, they put together 99 different studies. Each of those studies had said, we found a difference between people with depression and people without depression. But when they put them all together, they found nothing consistent between them. So 99 studies all said there was something going on, but when you put them together, you find nothing. That seems like a problem, right? So we think that one of the reasons for this has to do with what we call statistical power, which is basically how large is the sample that's going into the study. Statistical power means how likely are we to find an effect if it exists. And what we've known for over a decade now is that most studies, both in neuroscience specifically, which is shown on the left, and kind of across biomedical science, have very low statistical power. So if your study has, let's say that somebody does a study that has 10% statistical power, that means that 9 times out of 10, even if that effect exists, they're not going to find it. because they just don't have the power to find it. That's in medical research, that's called a futile study, and you wouldn't be allowed to run a clinical trial in medical research, but people regularly do basic research studies that are basically futile. I'm not going to make you read math, but one of the reasons that this really matters is that it's not just about whether you're going to find the thing, it's about how much you can trust the findings that come out of underpowered studies. And this analysis, John Ioannidis wrote a paper back in 2005 called "Why Most Published Research Results Are False," which I think is, and this is the basis of his argument, which is that if you run an underpowered study, not only are you likely to not find something if it exists, but the results that you do find are very likely to be false, much more likely to be false than if you had a highly powered study. And so this means that it's not just that we don't find things, we also can't trust the things that are found by those studies. Here's an example. So back around 2000, it became possible to start looking at the versions of different genetic variants that people have. There's a lot of variants that are pretty common across people. And so a bunch of studies started asking, how does brain structure or brain function differ between people with, say, two different versions of a particular gene? This is an analysis of a bunch of studies that looked at differences in a particular gene for this thing called BDNF. And they were looking at the size of the hippocampus, which is really important for memory. So what this shows is on the bottom axis is basically when was it published. The y-axis is how big was the effect that was reported. And then the size of the circle is how big was the study, how large was the sample. And so what you see is early on there were studies with reporting very large effects with very small sample sizes. Over time, the sample sizes get bigger, the effect sizes get smaller, and here it looks like it goes to 0. And in fact, we know that it goes to 0 because in genetics, more than 10 years ago, people moved to deciding that we really need very large samples if we want to understand the association between particular genes and particular phenotypes. And so when people started doing these studies with tens of thousands of subjects, they found that if you look at what genetic differences are associated with differences in the size of the hippocampus, none of the ones that people had talked about before in those small candidate gene studies actually replicated. They found new genes that nobody had ever heard of that were related to hippocampal size, but none of those ones that people had talked about were. And that suggests that people spent a decade at least fooling themselves about the relationship between those genes and things going on in the brain. So how well-powered are fMRI studies? We did an analysis back in 2017, and this is an updated version, just looking at statistical power and how it's changed over time. And so what you see on the left is the sample sizes of fMRI studies that we kind of extracted automatically from text. That's a log scale, so it's actually going up more than it looks like. But the median study in 2023 had 33 subjects per group. And so the question is, how good is that? What kind of effect can one find? And we did some analyses to suggest that this could find like a blob in the brain with an effect size of 0.69. That's a pretty big effect. In the realm of effect sizes, that's almost it's kind of medium large. Now, we'd like to know if that's a realistic effect, because you want your study to be powered to find a realistic effect. So, for example, if I told you, I'm going to go, I want to do a study to find differences between in height between 3-year-olds and 20-year-olds. You don't need a very big sample to find that, right? You could find that huge effect with a very small sample. If I tell you I want to find the difference in height between 18-year-olds and 19-year-olds, there you need a much larger sample because people overlap a lot more. So that's the idea here. We need to make sure that we're powered to find a realistic sample size. So we came up with a way to do that based on openly available data, where we took the data from people doing a bunch of different tasks in fMRI. We knew about the region. We needed to know where do we look in the brain to tell what's the effect size. So we both took the regions that this particular study, the Human Connectome Project, had suggested would be involved in each task, and then we used a system that we developed called Neuroscience, which is a large databasing project, to let you find, for any particular term, what regions in the brain are associated with that term. So we find regions where we're pretty sure that they should be active, for example, in a motor task. And then we looked at how big are the effect sizes. On the left, you're seeing what we call the percent signal change. This is how much the signal in functional MRI goes up within a person. The biggest you'd ever expect that to be is about 4 or 5%. And that would be in, if I make you move your hand versus doing nothing, or if I shine, flashing stimuli versus doing nothing and look in your visual system. And some of these tasks were, like the motor task was getting up there around 4% signal change. So these are really big effects. But those aren't the effects that we care about when we analyze the fMRI. What we care about is how big is that effect with respect to how much it varies across people. That's what the effect size on the right tells us about. And the black line there is the line for where the median study in 2023 was powered. So what that says is if the effect is bigger than that, the median study had, so 50% of studies had enough power to find anything to the right of that line. The rest of them did not. And what you can see is even for these huge effects, only one of them is actually bigger than, like people moving their hand versus doing nothing. That's the only one, that's like a whopping signal. That's the only one that most studies were powered to find. So what this tells us is most fMRI studies are really badly powered to find any kind of interesting effect. Like these are not an effect that's as big as you moving your hand versus doing nothing. That's whopping. Most of the stuff that anybody who does fMRI in this room studies, the effects are probably an order of magnitude smaller than that, so much smaller. If we look at those depression studies, I don't know if you can see this, I've colored in red the ones that I'm calling having a grossly insufficient sample size. And this is based on, this is from a few years ago. There's a great paper, it was called False Positive Psychology, which was about all the reproducibility problems in psychology. And they basically said, you should never run a study with less than 20 subjects per cell, because no power analysis is ever going to tell you that that's an okay thing to do for a reasonable effect. And so if you're doing that, you're probably doing it for reasons that are questionable. And you can see that the majority of those depression studies actually had a grossly insufficient N. And that's, we don't know. I think that's probably why those results have been so hard to reproduce. So we think that the generalizability, ultimately, of neuroimaging results is imperiled by these small sample sizes. So how do we fix it? I don't want to get up and give a talk and just tell you how everything's broken. I want to tell you how we and others have been trying to fix it. This is work that others have done. We haven't been involved in this. This is something called the Enigma. The study that I showed you with the 20,000 people that showed us that the candidate genes didn't work, that was done using the data from this Enigma project, which has been led by Paul Thompson from UCLA, which is basically bringing together a bunch of groups to amass very large sample sizes. We've taken a different route, which is to try to enable researchers to share their own data via a platform we developed called Open Neuro, which is a data sharing platform. It's one of, if you've heard of the BRAIN Initiative, it's a large federal project. This is one of the official data archives of the BRAIN Initiative. So anybody who's funded by the BRAIN Initiative has to put their functional MRI data in this archive. Now, sharing data is not that hard. Sharing data well is really hard. And I'm not going to play this video, but there's a funny video about data management. I know, funny video about data management. You have to be kind of a geek. Data management, data sharing and management stuff. And it's basically about all the problems that people have when they try to get data from people and reuse them. So we started working almost 10 years ago now to develop basically a way to organize data so that we could just hand the data to people and they would know what to do with them without having to ask us any questions. Because everything they need to use the data would be built into the data. It's this thing we call the brain imaging data structure. And it basically it kind of does the things that people already were doing in their lab, but it kind of formalizes them and codifies them. And it provides us a way to look at a data set. We have this piece of software we call a validator that can look at a data set before anybody ever uploads it and tell them, does it actually meet the standard? Is it organized in the way that it's supposed to be organized? And tells them how to fix it if it isn't. I sometimes jokingly refer to this as plumbing for science, because it's like hard work that it just, like, you know, nobody really thinks about, right? It's like telling people how to name their files and organize their folders and put their data into the files. But this took, like, you know, thousands of hours of work by hundreds of people. This is the timeline of all the meetings that we had, all the papers that came out, and this has now been extended to a bunch of, we started with MRI, but it's been extended to a bunch of other data types, and it's been really successful. We had a paper that just came out that I wrote, The Past, Present, and Future of BIDS, with over 100 authors, people who've been involved in the project. And this is just showing the number of data sets. This is an underestimate for sure, because this is just one database that tracks whenever people process BIDS data using this tool we've developed called MRI-QC, I'll talk about later. But we can see that there's, as of at some point last year, there were more than 400,000 functional MRI data sets and more than 300,000 structural MRI data sets that had been converted and run through this particular piece of software. So the real numbers are almost certainly much bigger than that. So lots of data out there, because people have really bought into the utility of having data that are well organized. Okay, so we talked about generalizability. Let's move to robustness now, right? So this is same data, different analysis. And one of the ideas that people have realized recently, that it's often the case that if you give a bunch of researchers the same data, they will not always come up with the same answer. This has come to be called multiverse analysis. The idea is that there's a multiverse of different analyses one might do. And John Ioannidis has a great term, the vibration of effects. The idea is, like, you know, you do different things and the effects kind of move around. They vibrate. We know that this is a problem in MRI. This is an example of, just from one software package, all the different choices that one can make. And they're all, like, each of these would be a reasonable choice. So if you put all those together, there's almost 70,000 possible workflows that one could come up with, none of which would anybody look at and say, wow, you shouldn't do that, right? They all seem like reasonable things to do. And we know from Josh Karp's work that if you look at the literature, people actually do use different methods. So he found that there were he looked at 241 papers and found that there were nearly as many different ways of analyzing the data used across those papers as there were papers. He also did this, this was done back in 2012. He was like a real pioneer, where he took one data set and ran it through almost 7,000 different analysis pipelines. He did this when he was a graduate student. And he found that there was a huge range of results between the highest and lowest. He basically just said, what's the range of the statistics from highest to lowest in terms of standard deviations? Sometimes you would get 8 standard deviations difference between the highest and the lowest. That's a lot bigger than that's probably on the order of the difference between a 3-year-old and an 18-year-old in terms of height. Really big difference. So how do we address this? Well, one way we can address it, one thing that we think has happened in the past is that people would often try a bunch of analyses and then report the one that looked the best, right? We call that p-hacking. And so one idea to address that is what we call pre-registration, which is that you basically write down what you're going to do and put it in a place where it's immutable, like there's platforms like the Open Science Framework, so that then people know what you were planning to do before you did it. And you also know, it's easy to kind of convince yourself, oh, I really meant to do that, when 6 months ago or a year ago you didn't. People sometimes worry that this prevents you from exploring your data. It doesn't. It just makes sure that you know which things were really exploratory and which things did you really hypothesize a year ago. This had an amazing effect in medical research. This is a paper by Kaplan and Irvin that looked at what happened. So the National Heart, Lung, and Blood Institute in the year 2000 started requiring that all clinical trials be registered through this thing called clinicaltrials.gov. And they had to say, in particular, what was their primary endpoint? Like, you know, are you trying to reduce, let's say you're giving people a drug, you're trying to reduce like number of heart attacks or blood pressure or number of deaths, like what are you trying to fix? The line with the flag to the left of that is what we saw before pre-registration was required. Everything, each of these little pluses is a positive, paper that reported a positive outcome from the clinical trial. The majority of clinical trials reported positive outcomes. 2,000 pre-registration is required. Suddenly, most clinical trials are reporting null effects, very few positives in comparison. It's not as if drugs stopped working in the year 2000, right? What stopped happening is people couldn't move the goalposts anymore and find an endpoint such that they could say that their treatment worked. Now this can help fix the kind of p-hacking problem, but what if, you know, you just had different people whose labs use different methods? How much does that matter? So we did a study. We wanted to know what's the effect of these different analyses when you remove any kind of incentives to find a result. So we did a study with a bunch of other people, really led by Tom Schoenberg and his grad student Rotem at Tel Aviv University, to basically try to ask this question. So we collected data in Tel Aviv on 108 subjects doing cognitive tasks, the details don't matter, where people made decisions, and then we basically gave those data out to groups of researchers and said, we'd like you to analyze these data like they were your own. Like pretend that these are data that were collected in your lab. We're going to give you a set of hypotheses to test that are of the form, is there activity for some particular aspect of the task in some particular brain region? Is there, you know, so for example, people made decisions about different levels of money they could win. Is there, you know, a difference, is there a relationship between activity in this part of the prefrontal cortex and the amount that one could win? And we gave them about 3 months to do that. And they had to sign a non-disclosure agreement because we didn't want them telling other groups what they had found. And they were all offered co-authorship on the paper, which is why the paper had over 200 authors on it. Don't try to write a paper with 200 authors. It is not fun. And we told them basically analyze it as if it were your own data set. We gave the data out to 82 teams. We actually got data back from 70, which was pretty surprising, because this is like many, you know, tens if not hundreds of hours of work. And the teams told us what they found. They also gave us a bunch of additional information that I'll mention in a moment. So we had them give us a really detailed description of what they had done. And amazingly, even with this 10 to 15 page description of what they had done, we often still couldn't tell what they had done. We had to ask them for computer code or files. But none of those 70 teams used the same analysis workflow. So 70 different analysis approaches. So here's the finding. Let me explain this for you. The x-axis, those are the 9 different hypotheses ordered from lowest agreement or lowest likelihood of saying yes to highest. So and then the y-axis is what proportion of teams said, yes, there is an effect. So if everybody agreed, those numbers would all be 0 or 1. Either everybody would say, no, there's no activation there, or everybody would say, yes, there is. For 3 of them, you see it's about 5%. So that's pretty much everybody agrees there's no activation for those 3 hypotheses. Pretty much everybody agrees there is activation for 1 of them. And then the others were from 20 to 40%. So given it's the same data, that's a really distressing amount of variability. And so that means across teams, there were 33 different patterns of outcomes. We also see a lot of variability if we just look across the whole brain at what areas, at each spot in the brain, what proportion of teams said there was activation in that area. The best that it gets is around 76%. So there was no place in the brain where even 90% of teams said there is an activation area here. And this is for the best one. It gets worse from there. This is a really interesting thing. So we dug into the actual, rather than just looking at how they answered the yes-no questions, we took the whole brain data that they gave us, like at each voxel in the brain, how much of a response is there? And we looked at the correlation between those. So this big, all you need to know here is this big red blob up in the top left. This is a correlation matrix. This basically says that for about 50 of the teams, their results were pretty similar to one another at the whole brain level, even if their yes-no answers weren't. So there's something about going from a whole brain pattern of activity to a yes-no answer that caused them to differ, and we can speculate about what those are. But you'll notice this group right here, these 8 teams, and this is blue here because it means that their results are actually negatively correlated. They're opposite of what all the other teams have found. And you can actually see that here. You know, like it's blue right here because it's negative, it's red here because it's positive. And so they found the opposite of what those other teams found. And we had to dig in. It took a lot of work to figure out what they had done wrong, but it turns out that they had basically set up their statistical model incorrectly in a way that is not probably that uncommon, but it takes there's a whole other talk I have about like the details of modeling in fMRI and how you really have to get it right. Otherwise you can end up with this. Nonetheless, we can put everything together. We can take all the 70 teams, even the ones that got it wrong, we can take them, put them all together, and we still get overall answers that are reasonable, actually fit with what we knew before and what we would have expected. And so we've now moved to thinking about applying this kind of multiverse analysis at scale. It's hard because each of these analyses requires a lot of time. If you want to do it 100 times with 100 different analysis pipelines, that's a lot of computing. And so we've put in a lot of work in the last 5 years or so building software to help actually orchestrate this type of analysis using large-scale supercomputing. We actually still do a lot of supercomputing in Austin because they have an amazing supercomputing center. And we're trying to make this available to people more broadly because we think that we do need to move away from thinking that there's one right analysis to do to thinking we want to understand how variable are the results across different analyses. We've started doing this. This is one recent example that I just wanted to kind of show. We've been really interested in this concept called reliability, which is basically if you do the same scan on the same person multiple times, how similar, and you measure quantitatively how much activity is there in a particular area, how similar is that across the 2 times? And we actually know that this is distressingly bad. This was a paper published a few years ago looking at a couple of large studies. You'd like these numbers, these reliability coefficients to be at least about 0.8. And in general for fMRI, they're pretty much never at 0.8. They're usually around, the average is about 0.4. This is important if you want to look at differences between groups or correlations between people and other measures. You really need a reliable fMRI measurement. So Michael Dimodinko in my group wanted to know what happens if you look at how reliability varies across different analysis pipelines. So he took 3 different data sets, focusing on a task that I'm not going to talk about today, which looks at kind of like reward processing. And he basically asked for each of these, how reliable are the measurements? And how does it differ across pipelines? And I'm not going to go into the details about this, but what I want to just highlight, this is the line up there is basically ordered. This is the reliability of different analysis pipelines on the same data, ordered from smallest to biggest. So what it says is that, like, at worst, reliability is basically 0, which means that you should not try to compute a correlation with those results. At best, it's a little bit above 0.4, which is still not particularly good, but it varies a lot. The way that you set up the model, particularly what matters is the way you set up the statistical model. The way that you pre-process the data doesn't quite matter as much. So this just highlights the need to understand not just one answer, but really the answer across lots of different pipelines. So we've also been, we've been trying to build tools to help people do better analysis because we, having come from labs where, all the analyses were kind of built by graduate students and postdocs and kind of held together with duct tape and nobody quite knew how they worked, but they just kept using them. We wanted to build, take what we know from software engineering about how to build really robust software and bring that to functional MRI. And so we've developed, this is a whole kind of like ecosystem of software, some of which is from our lab, some of which is from other labs. It's an open source software ecosystem, so everybody shares. I'm going to focus on one particular thing that we developed, which really grew out of an idea. Chris Gorgolewski was kind of the person who worked with me to develop this BIDS framework. And he had this idea that one thing we could do, once we know the structure of the data, then we can build software tools that could just look at the data and you don't have to tell it anything, right? They're like, right now, a lot of software tools, if you want to use them, you have to like get the software, install it, and then reorganize your data so that it'll work with the software. He developed this idea of what we call a BIDS app that could just look at a data set and automatically know how to process it because that data set follows this BIDS organization. The first one of these that we developed is this thing called MRI-QC, which is a quality control package. So quality control, you would think that, oh, everybody does quality control on their MRI data, right? And they try, but there haven't in the past been really solid tools for looking at data and seeing, what's problematic with the data. Are there particular individuals whose data is messed up and so on? So Oscar, who's now in Switzerland, he was in my group when he developed this, developed MRI-QC that basically can take a BIDS data set and very easily create a report that can show you, are there outliers, and then you can dig into individuals and see what might be going on to really give you a feel for the quality of a data set. And all you have to do is put your data into the BIDS standard, and then you can just very easily apply this. The other thing that we've developed that's been really successful is this thing called fMRI prep, which is when we get MRI data off the scanner, before we can do our statistical analyses, there's all this stuff that has to be done. This is like the flow chart of all the things, right? And then in general, this can take anywhere from like 8 to 24 hours, depending on the size and the complexity of the data set. And in the past, every lab kind of did it a different way, and they would like use one particular software package, even though it didn't do everything the best way, because working between software packages was too hard. So we came up with a way to basically put together the best parts of each software package to try to do a better job and a more robust job of pre-processing. This is the usage of this software that we track. Whenever anybody runs it, unless they opt out, it basically sends a little message home, and particularly tells us, did it crash? So we know not just how many people are trying to use it, but how often is it actually working. And we see really consistently it's about 68, 69% successful, which doesn't sound great, but it's actually for like research software, it's pretty good. And the other thing you can see is that it's being used, this is per week, right? So it's being used successfully 6,700 times per week over the last basically year and a half. So I think for academic software, that's pretty successful. And so we're currently funded by the Brain Initiative to continue developing this and hope to keep doing so. All right, so talked about how do we try to make things more robust. Are they replicable? Well, I'm going to focus in particular on what does it mean to run the same analysis, because there's, a lot of people think about like, what does it mean to go run the same experiment? There's less attention to what does it mean to run the same analysis. And I mentioned that in that NARPS project, we often couldn't tell what people had done. For those 7 teams that did it wrong, we had to go back and get a bunch of files from them and computer code to figure out what had they actually done wrong, because it wasn't clear from that description. So we need to be sharing more than just a written description. And I think that I'm inspired by this quote from a while ago by my colleague David Donahoe. An article about a computational result is advertising, not scholarship. The scholarship is the full software environment code and data that produce the result. So we really need to get beyond just thinking the paper describes it. The research is really described by all the objects, including the code and the data that went into to doing the science. I'm going to show you an example of how this has helped us. So we had a paper we had put up on, so now, I don't know how many of you are familiar with archives like bioRxiv. Now it's common for, whenever we want to publish a paper, the first thing we do is we post it to one of these preprint servers like bioRxiv. And in general, we post it with the code and either sharing the data or, in some cases, the data are already available. So this was a paper we had posted on bioRxiv that was looking at some issues with this large study, the ABCD study. Those data were already available, and we had kind of pointed out some challenges. And so we put it up, and then, and this is something we had said, for 1,326 of the 7,231 subjects, this particular thing happened that is problematic. Well, one of the ABCD programmers dug into our code and said, oh, you made an error, right? Sage found this error, and it was like if you write code, you know that this is like a horribly structured piece of code, right? Basically the problem was one parenthesis was in the wrong place and it was improperly grouping things. It's a terrible thing to have done. And it caused us to make this error where we weren't indexing things correctly. The answer turned out to be 447, 6.2% rather than 18.3%. Didn't really change the overall message of the paper. The paper was ultimately published in eLife. But it helped us get it right. If we hadn't put this out before we had submitted the paper, we wouldn't have known that, or we might have had to issue a correction to the paper after it was published. But sharing the code and the data up front allowed this to be found by people in the community, and we thank them for it. Like, if there are errors in the code, we want them to be found. We don't want to publish things that are wrong. Many people are scared of sharing their code for lots of reasons, right? And I think in part, one of the reasons that I like to show this example of a mistake that we made is that, I think it's important that we normalize errors. We're humans. Humans make errors in everything that we do. We want to try to reduce those as much as possible. And one of the ways to do that is have more eyes on it, right? Code that's looked at by 5 people is going to be much more robust than code that was looked at by 1 person. And so I think this piece by Nick Bartz from a while ago does a nice job of arguing that basically you should be sharing your code in part because if it's not good enough to share, then why do you think it's good enough to write a paper and submit the results based on those code? So we've now moved towards sharing all the code for all of our projects, and I think it's really important for this sort of the ability to reproduce analyses. Finally, I want to talk about this reproducible idea: same analysis, same data. This sounds like that should be easy, right? Turns out that it's not. My colleague Mike Frank and his group looked at data from 25 different articles published in this journal, Psychological Science, which gives you a badge if you had basically made your data available, open data badge. And they initially found, they ran the analyses that were described in the paper on the data, or if the code is available, they ran that. They initially found that 64% of the articles had at least one major discrepancy in the results, at least a 10% difference between the published result and what they found. So then they went back to the authors to say, can you help us figure out what's going on here? And ultimately they were able to get the same results as the original authors, 36% of the time without talking to the authors at all, 24% of the time after talking to the authors, 12% of the time with no response from the authors. They basically couldn't reproduce it and they didn't get any help from the authors. 28% of the time, even with the authors' help, the authors could not tell them how they could get the original result again. A quarter of the time, right? And these are presumably the best because these are people who are actually sharing their data. So it just highlights how hard it is to really computationally reproduce. I want to show an example of a particular challenge that seems like it's like so deep in the weeds that it shouldn't matter, but it does. So I did a study back when I was at UT Austin. I did a study where I got into an MRI scanner about 105 times over the course of a year and a half to try to basically look at how brain function changed over time. And I had blood taken to do RNA sequencing, like lots of measurements. And we published a paper in 2015, and this was, I had gotten the reproducibility bug at this point. So we also published this thing we call a virtual machine, which is basically like a little computer within a computer. So you could download this thing, run it on your machine, on your Mac or your PC or whatever, and it would reproduce all the analyses, it would pull down the data and the code and reproduce all the analyses that we had reported in the paper. And so we thought we were great, right? Nobody does this. And I got an email. Hi, I'm an undergraduate student at Johns Hopkins. I'm working with your data. I've run into some trouble reproducing the RNA-seq WGCNA results. There's supposed to be 63 modules, as in the paper, but when I run the scripts, it gives me only 60. I also reran it with the VM, and the results are only 60. Essentially, the same code is run on the same data, but arrives at 2 different results. I've been scratching my head at why this may happen. And then this person said, I noticed that this R package, the software package we use for these analyses, has been updated. Maybe this is causing the issue. And it turns out that this was the problem. We had done, this highlights the fact that you should always put the version of the software you're using because that's important. We had used version 1.47, and by the time this person was doing their analyses, it was version 1.51. That doesn't seem like it would be such a big difference, but it changed the results. It just highlights how hard it is to really, especially when you have complicated analyses, to really reproduce the results. We actually know from neuroimaging that that there are some kind of distressing differences that can rely. This is, FreeSurfer is a commonly used package for measuring like the thickness of the cortex from MRI. This is the same version of FreeSurfer run on 2 different versions of the Linux operating system, which is one of the most commonly used systems that people use. And you can see differences of a couple of standard deviations in thickness estimates in particular places. You would really hope that the different version, the software is exactly the same version. It's just like the underlying system libraries that differ, and those are giving rise to these differences. And you can also see it with different versions of the software as well. So one of the things that we've moved toward is something called containerization. There's various technologies. We use a thing called Docker. But the idea is that instead of just sharing your code and your data, you share this entire container, which is sort of like a little computer. And it has unlike what we did before with the virtual machine, we make sure that it uses exactly the same versions of all of the software packages. That's gotten easier now across different platforms. This helps. It's not perfect, but it does help to some degree with reproducibility. And I'll just highlight how, all of the BIDS apps that I talked about, like fMRI Prep, are built in these containers. The reason I'm showing this is that this is one line that one could write in your computer to run fMRI Prep. You don't have to download anything, right? All you have to do is install this Docker software. It will go pull the specific version of fMRI Prep you're asking for, 20.2.0. And then it'll get your data, and it'll run that version of fMRI Prep on your data. And if you go run it again, it's as close to fully reproducible as you're ever going to be able to get. It may not be perfect. If there's any kind of random numbers anywhere, then things are not going to be perfect. But this is as close as we can get to kind of really robust reproducibility. Okay, so just to sum up, we have problems. I hope I've convinced you we have problems. I think we have solutions. I've laid out some of them around data and code sharing, containerization, building larger samples, building consortia, data sharing, multiverse analysis, using really robust tools. Ultimately, I think the problems, the technical problems are always easier than the social problems and the incentive problems, right? That right now the problem is like people are incentivized to get papers in fancy journals, and actually, that pushes you to tell a really clean story, whereas, the real story is almost always certainly going to be messier than the really clean story. And getting people to buy into the need to do this stuff is the hard part. The technical stuff, I think we're well along the way of making it happen. Just finally want to thank lots of people who've worked with us on this, lots of people who've given us money, and thanks for the invitation.
Okay, great. Another reason for struggling scientists to lose sleep at night. And so, questions for Russ?
Do you think AI would help with the algorithms and call data more easily?
Do I think that AI would help? Yes. So there almost certainly are ways in which AI will help. One, so we've actually been doing some work taking advantage of this open neuro data set, which has thousands of people's worth of data. We think that we can, if we train, in the same way that, like GPT-3 or GPT-4 were pre-trained and then used for ChatGPT, right? And ChatGPT could do all these sort of things, even though GPT-4 was just trained to say what the next word is, right? So the idea is that you can pre-train a model on a bunch of data, on a very large amount of data, and then adapt that model to doing other things, be it, writing a poem or, answering questions or whatever that might be. So we've been doing some work trying to build what we call foundation models for fMRI data, where we train a large model to just predict the next time point in the fMRI, and then use it to do other things. And we've actually found that you could do a much better, if you use that kind of pre-trained model, you can get by with smaller data sets than you could have if you were just kind of training the model to, say, predict, what a person's doing, if you're just training it on the data themselves. So I think that's one way in which AI models, kind of inspired by models like the ones behind ChatGPT, can actually be helpful for this. But it's still really early days.
Okay, other questions?
I'm a software developer, and when you show that code there that, oh, here's the error because the expression was too complex and thing, and one of the major problems that I found when I'm writing code is not the code per se, but is the definition of the problem that is faulty. And then what are you guys doing in terms of verifying the quality of the hypothesis or the definition of the problem so we can write more precise code to represent that?
That's a much harder problem, right? Because you have to deeply understand what's going on. You don't have to deeply understand anything to write a test to make sure your software, you have to deeply understand the software, but not the data, right? But there, you really need to understand the scientific problem and the data. And so some of the work, like the project that I talked about looking at reliability, is starting to get at that because it's looking at, different ways that one can specify the model. But ultimately, I think the best way to get at what you're talking about is to have either simulated data, where you know the answer. Actually, I think that's the best way. Like if we had a way to simulate functional MRI data where I put the signal in and then I ask, can my software get it back out? That, I think, is the best way to do it. Unfortunately, we don't really have great simulators for functional MRI data right now. There's some that are coming around, but it still doesn't work that well.
Okay, lots of hands going up there. Oh, Steve. Oh, Steve again.
I have the mic. When I look back at my pathology imaging days and where, you leveraged AI to do things people don't do well to enhance the overall, is that where we are? Because it seems to me there's two buckets of problems. One is documenting kind of what you did, and the other is there too much variability in what's happening inside the computer in terms of whatever software they're using? And is there going to be a move to standardize that, or at least standardize the format of the imaging?
Yeah. I'm wary of saying that we should standardize the way that scientists ask questions, simply because that could end up, kind of restricting our ability to find interesting new things. Because we almost certainly don't know all the right answers right now. So that's why we focused more on standardizing the data so that anybody can take them and kind of ask different questions with them. But I think we do want to know, to the degree that there are better and worse ways to preprocess your data, we should know those. We don't. It's kind of distressing that we don't know those right now, even though people have been doing this for a long time.
Oh, right over here, this young, fresh face.
So I'm a junior in high school, and I assist a student. He's a senior, and he has autism, but he also suffers from depression. So how do you think with his patterns of thinking and his brain, I guess you would say, how do you think that would affect your research that you did with the whole depression and?
Yeah, there's a lot of interesting layers there, right? Because one, I think there's one of the reasons that the depression research probably is less consistent than you would hope may have nothing to do with brain imaging, right? It has to do with the fact that a diagnosis of depression is not a biologically coherent phenotype, right? It's like you can have depression for a lot of different reasons in a lot of different ways. It's basically a checklist that a physician uses to for insurance purposes, right? And so there's been a lot of, in the NIMH, there's been development of this thing over the last, I guess, 15 years now called the Research Domain Criteria, RDoC, which is trying to get at this issue of what are the underlying dimensions? Because we also know that, if you look at the genetics findings, there's a ton of overlap between, say, autism and depression and ADHD and bipolar disorder in terms of, what genetic differences are associated with those those outcomes. And so I think that, that's probably one of the biggest challenges, right, to answering that kind of question is like, what do we, how do we reconceptualize these diagnoses in a way that does a better job of, of relating them to biology? Because clearly right now they're not cleanly related to biology.
So what is a way to ask a question if I'm invited to be in a clinical trial? What kind of question could a layperson ask of the people who are doing the study? That's
a really great question. One, have you pre-registered the trial, and can you tell me what are the endpoints you're looking at? Beyond that, I think it gets deep into the weeds of what the particular trial is about. But I think just asking them, making sure that they have, one, pre-registered it and can show you if it's a federally funded trial, then they would pre-register it using this clinicaltrials.gov website, where you can just go and search for clinical trials on, whatever disease of interest. I wish I had more kind of general purpose sort of things to say, but beyond making sure that they've thought about the study being well powered and that they've, done a pre-registration, I think then it just becomes much more kind of like study specific.
Okay, next question.
Wonderful talk. Thank you so much. I've noticed a lot of what you talked about was making sure that the data are reproducible post-acquisition. And I think one thing, and to ask you, how much does it matter if different universities are using different scanners, different head coils, different sequences? So how can we address that area of variability?
Yeah, that's a good question. And we know it makes a difference. We've known for a while people have looked at differences across scanners. We know from the ABCD study, which is collected across a bunch of different sites, that even the same make and model of scanner can show differences between sites that may have to do with the humidity in the scanner room or various things like that. There are methods that people have developed, Combat being the best known one, to try to kind of address these differences. Not clear that they actually work. I have a postdoc in my lab right now, Eric Bridgeford, who's working on novel methods for addressing these, what we call batch effects or site effects. It's far from a solved problem right now. I mean, we're not the only people who suffer from this problem. Like genomics, has been dealing with these kind of batch effects for a long time. So part of what he's doing is trying to bring to bear what's been learned there, because the simple models like this Combat thing seem to be problematic and could sometimes actually cause bigger problems than they solve.
Okay, Sandy.
So Russ, elegant, very sophisticated, whoa, on so many levels. My question to you is, and I know it may be a bigger answer than you can give, but I want you to help us think about it with Jeff. As we're with the Brain Health Project, we're looking at individuals being their own comparison versus, what happens across this group data, and we're interested in change the brain. How does the brain improve as people get certain things? What do you feel like might be our most promising ways to get it right at that level with imaging? If you were going to partner with us as a collaborator, what would you have us do?
Collect more data per person.
We have lots of data collected.
So I guess the question, do you, at each time point, collect a lot? So I don't know how much, that's the question is, how much do you have at each time point? For any particular MRI measurement in particular. So let's say you want to look at resting fMRI. We used to think that 10 minutes of resting fMRI was enough to compute something reliable. We now know that you probably need at least 30 minutes, if not more, to get reliable estimates of brain connectivity. So that's the direction that my lab has gone. Is we now, for most of our studies, we're collecting like 10 hours per person over the course of months just for feasibility. And that's not even to look at longitudinal change. That's just to look at like brain organization within a person.
Bless those people.
I know.
Because an hour, I mean, it's hard to get
it is.
people in those tight scanners for it. So ours do it at baseline 6 months and then 1 year every, follow it up from there. But and it's an hour at each time.
Yeah,
but
It is hard, yeah.
But 10 hours each time, we might lose most of them.
Right. I don't think you need 10 hours each time, right? But I think you need, my guess is you need more than you're getting. If what you're getting is based on what people have done over the last decade, you probably need more than you're getting.
You sound just like a biostatistician.
I hang out with them a bit too often.
I'm greedy. I need more data.
I know. Yeah.
Okay, Benton Pierce has it.
Yes, Russ, back to the reliability issue. So we know that, especially as we get older, there's intra-individual reliability and behavioral performance. And I guess, I hope this isn't an off-the-wall question, but how does that map onto variability within people in terms of brain activation?
Really good question that I don't think we know the answer to fully. It depends on what time scale you're looking at. So like in that my connectome study that I did on myself, we had resting state data twice a week over the course of a year and a half. And there were some days that, one, we had a manipulation because half the time I was like caffeinated and fed and half the time I was not caffeinated and unfed. And you actually see, big differences in like resting state brain organization there. Or even if you just look at, I had done like ratings of how tired I was. And you certainly see differences in resting state organization related to that as well that are over and above just what you see from caffeine versus not. But I think in general, especially for task fMRI, I think we just don't know because there's also the overlay there of, as people do a task, they're going to change, right? They're going to get better at it. And so the question is, how do we address those differences and then understand whatever other variability might be going on?
So are these results sort of pointing us towards studying the variability, say, between individuals and
within? I think we want to understand both within and between. Yes, right. But the problem is we need, I think what we've, kind of depressingly, what we've learned is like if you want to do a good job of looking at individual differences, especially like brain behavior correlation. So there's this, kind of splashy Nature paper, I guess a couple years ago now from Marek et al., which basically suggested to look at correlations between like brain connectivity and behavior, you need thousands of people. Just like we know you need thousands of people to find correlations between individual genetic variants and behavior. But then you also, if you want to look at an intra-individual difference, you need a lot of data within each person. So nobody's going to go collect 10,000 people 10 hours each, right? That's just that's infeasible.
Yes. So with all this, what is the role, what is the place for really novel questions where the funding is probably going to come from sources that aren't going to allow many subjects over long periods of time? How do we think about those kinds of studies?
We hopefully think that they're really important. I worry that, so many people have become like accustomed, in my lab too, we've become accustomed to using data off the shelf to ask a lot of questions. And I think that the art of developing experiments is actually kind of waning in a lot of, cognitive neuroscience circles as people do this. And so I have some concern that that's going to kind of interfere with the ability for people to know how to develop those new studies. And then, you have the challenges of convincing the funding agencies to fund them when they've become so focused on these very large studies.
It seemed a bit rhetorical. Oh, I'm sorry. This is an actual question. Okay. What is the duration of conditions under the use of different software for measurement? Just what it says. Go with that.
I'm not sure how to parse that.
Let me see if I can formulate it in some way. What's the duration of conditions? Well, I guess they're asking how much data do you need for a given condition in an experimental task? Right.
I think the answer is it depends, right? It depends on the size of the, the amount of data that you need always depends on the size of the signal you're looking for. Going back to my 3-year-old versus 18-year-old height issue, right? And so if you're looking at like activity in the visual cortex for flashing stimuli, you don't need much. If you're looking for something very subtle around language or emotion or something like that, you might need a lot more.
Okay, with that, I think we're at time, and let's give Russ a big round of applause for all the great issues that he's raised and is really addressing in such amazing ways and devoting so much effort to it. Yeah. Thank you so much, Russ.
Thank you.
Lifelong brain health. This is our shared mission. Thanks to Russ, and thanks to our audience, for everyone for coming. And we look forward to seeing you again on April 5th. Dr. Carolyn Leaf will present on effectiveness of pharmacological interventions as assessed using psychoneurobiological approaches to mental health. See you then.